Thank you all for your time and listening, and hopefully this will be interesting to you. So a couple of months ago, Rucheng asked if I could speak about the adaptability of the mPrep system and our ASP automation. And I said I would, and I will concentrate on some novelty or somewhat unusual applications to show the potential, and it will also show the potential of my fellow electron microscopists to MacGyver solutions. So with that, I'm going to begin, and I will move fairly quickly because I'd like to show a variety of applications. There are some uh, uh, 35. Let me move a menu out of the way so I can see my slides here. There are some 35 or more TEM biological specimen applications that have been published or presented that I'm aware of using mPrep system, and about 15 or more for volume EM. These cover a very wide range. I should say that many applications that I know of have not been published mentioning the system, but they are certainly being used. So I just thought I would make that point here. And let me add a laser pointer, so I need that. So a wide range, bioresearch, clinical biopsies, a variety of tissues, breast cancer, pancreatic cancer, mesenchymal stem cell models, some non-mammalian samples, cells, viruses, cell pellets, etc., and quite a range of methods from volume EM through X-ray microscopy and even cell culture, and as well as some materials research. And let's see. So it was very difficult to choose, and that's why I'll move fairly quickly. For those who are not familiar with the system, I can show the basics here. The fundamental parts of the system are capsules that hold specimens and capsules that hold TEM grids. There's our automated specimen processors, the ASP 1000 and 2000. There's a device that's a 96 well plate made out of a malleable silicone rubber that seals the bottom of the capsules. The capsules have pores on the bottom on the specimen capsule, and there's a pore on the bottom of the grid capsule. So you can place them in here to hold the liquid in here. There's a device called the orientation workstation, and I'll show some examples of that. And there is a specimen and grid holder for critical point drying, and it's also a high thermal mass. It holds the capsules in these holes, and it can be used for some cryogenic applications as well. And I'll show some examples there. And the system is designed to be compatible with standard labware and related systems, so pipetters, micro well plates. The capsules fit into microtome chucks, and critical point dryers, and even cryovials for certain kinds of preparations. So, and again, I'm just going to review this quickly. A kind of a basic workflow is a specimen comes from whatever source, in this case, some generic rodent. It's placed into the specimen capsule. It can then be entrapped with the mPrep screen. These capsules attach to pipetters. And the pipetter draws the reagent into the capsule through these pores on the bottom for reagent exchanges and for certain kinds of agitation as well. The bottom of the capsule can be sealed while you cure the specimen for resin polymerization, for example, and that would use the mPrep bench, the silicone device I showed previously. The entire capsule can be chucked into a microtome to cut sections. Obviously, we don't see the mesa being formed here that one would do, and this limits the amount of specimen touches and provides some potential labeling and other opportunities. The sections, when they're prepared, are placed on grids, and these can be then placed in the mPrep G capsules, which also attach to pipetters. Now, I'm showing single channel pipetters. The reality is, in most applications, you would rather use a multi-channel pipetter if you're doing things manually, and I'll show some examples of that work, either with the specimen capsules or with the grid capsules. And of course, you can automate the entire system, and I'm showing an ASP 2000 and an ASP 1000 here. The 2000 has heated and cooled reagent plates. So I want to talk a little bit about the capability of adapting workflows with the capsules. So I already showed where a specimen is entrapped with a screen, a single specimen with a screen. One can also orient the specimens in the capsules. I won't show this in detail, but they can be pinched by the screen and held in an orientation in the capsule. And one can also load multiple specimens into a capsule, not use the screen, and cap this with a second capsule. So now the specimens are entrapped between the pores on the bottom of this capsule and the pores on the bottom of that capsule. When you want to do the reagent exchanges, you place the specimen into a basin, a microwell plate, or a similar device. You attach the pipetter to it, or this is done automatically with the ASPs. What I'm not showing in here is a filter that goes between these two, and the reagents are drawn in. And these lines illustrate that there are 37 flow streams that rapidly direct reagent flow to and through the specimen in a bidirectional manner that leads to very fast sample preparation speeds. There are also two embedding options. This gives users a lot of flexibility. One can embed directly in the mPrep capsules, as shown here, and cure them and then do the microtomy on them as well. I think I saw Phil Seifert is here. This is a picture from a poster that he presented that shows retina being sectioned in the microtome. One can also flat embed for multiple specimens by simply unloading them from the capsule assembly and placing them into a flat embedding mold here. So I'm going to show breeze through multiple applications, showing how the capsules can be used in various ways. This shows that one can stack multiple capsules containing specimens together and multiple grid capsules as well to do high throughput production. So this first example basically simply shows how this technology can be used in a small laboratory. In this case, this is Chip Dye's laboratory, Louis Dye's laboratory at NIH. And I want to illustrate that he was able to achieve complete infiltration of myelin in a 45-minute preparation with the ASP. And one can do that with relatively tough specimens, and the myosiatic nerve is sufficiently tough by using the fast-directed agitation, the near-zero carryover, and slow warming of the resin to decrease viscosity and facilitate its infiltration. And I'll try to show where these applications are published in most of these slides here. And this was published in Microscopy Today just a couple months ago here. Another example, and this was briefly presented in some detail by Noah Flint in BioEMTalks approaching a year ago now, half a year ago. And RUP Labs is a CAP, ISO, and CLIA certified national reference lab for pathology. And the methodology that he used and that his laboratory uses, and this is a relatively large lab, about a half a dozen people, seeing multiple specimens from patients every day, a dozen or more. 18-gauge renal biopsy segments are received at the lab. They're cut into small pieces, and then they're placed into stacked capsules. They're attached to the ASP 1000 in his particular lab, and it's roughly a 2-hour protocol, and then samples are flat embedded. And here's some examples of the kinds of things. So most of their work is with renal and muscle samples, but they have also do nerve and cilia. I won't talk more about that here. So Phil, here's your work. Phil worked with mouse eye posterior segments. These are a fragile sample, and they're relatively large for mPrep capsules. In this particular case, this section through the posterior cup shows that the specimen was about 3.5 millimeters across. The inside diameter of the capsule is about 4.5 millimeters, so relatively large. And everything is very well preserved here with the staining in here, and here's the TEM with excellent preservation of the outer segments, the pigment epithelium, the mitochondria, and such. So again, just showing one of the examples of how this can be done. Phil presented this with his colleagues at National Society for Histotechnology last year. Another example in here comes from the laboratory of Claudia Lopez, work by Aaron Stupinski that was published in Methods in Cell Biology. They used an ASP 2000, did the preparation in about 6 hours, and as you can see, a variety of samples, a variety of methods, serial block-face SEM, mouse tumors, compared to manual, and an array tomography approach as well. So I just want to show the breadth of what could be done here in terms of volume EM. And one thing I think is important to look at here is that this particular lab also compared different kinds of sample preparation methods, using ethanolic UA, for example, and then a faster preparation as well. And that's one of the advantages of this approach. With automation, it allows one to look at many different parameters. This is just to illustrate that another laboratory, Cleveland Clinic, Benson and Kidd, have used the ASP to do processing of brain and brain cortex. Their prep protocol is about 7.5 hours to resin curing, about 1 hour of hands-on effort. This is a small lab of 2 people, and in their own words, they require automation in order to address the workflow that they have. And they pointed out in one of their papers in here that artificial intelligence segmentation was enabled with the consistency of the ASP preparation. But this illustrates that these same samples and samples within the capsule can be imaged using X-ray microscopy. This is a relatively easy process. The entire specimen that's embedded in resin in the capsule was examined for X-ray. So one can do correlative microscopy with these samples and within the specimen capsules, which provides a nice amount of convenience and sample integrity, and to be able to do this correlation over a very wide spatial range in here. A particularly clever MacGyver-ish application was developed by Emily Benson. She used this orientation method here that I've shown, and in this particular case, 3 nerve fibers were pinched in the mPrep screen. This is diagrammatically showing the workstation, and also a thread, a fiduciary thread in there, so she can tell the difference between which of these nerve fibers is which. So they are loaded into the capsules, processed on the ASP, and then they're embedded in the capsule, and one can clearly see the fiduciary thread and the 3 different nerve fibers in here. Sections were then cut from these in an array tomography-like approach, but not strictly array tomography, although the imaging was simpler. 1 micron sections were placed on cover slips. They were urinal and lead stained on the cover slips, placed on copper tape, placed into the SEM, and with the SEM that can use low voltage, primarily backscatter imaging, to image all of these different axons from these different specimens in a detailed way. And then these can be auto-segmented and analyzed. And with about 30 samples per SEM array and 3 nerves per section, they can do about 90 nerve sections in one array of specimens that goes into the SEM. So they have many axons to count, and this is very important for their preclinical therapeutic testing of various agents. Another very clever application was a cell pellet application. In this particular case, a cell pellet of lymphocytes. These were agarose embedded and ASP processed. And here we see the B cell pellet imaged with serial block face SEM, and then an individual B cell in this image stack here. And then this particular group in this study, of which Cleveland Clinic were co-authors on, did segmentation and analysis of mitochondria and the nuclei in here, and were able to obtain a lot of information on how mitochondrial volume changes relative to obesity. Obviously, I'm not going to go into the science here. This is not my work. Another application that was quite novel and somewhat MacGyverish as well came out of Kurt Simick's work at the Donald Danforth, and they did a cryo high-pressure freezing free substitution in the capsules. Here diagrammatically, the planchettes are shown in here, and they do light microscopy, X-ray microscopy, and serial block face. So this comes from their paper in here. They're using the CPD cryo holder in here, surrounded by dryerite to keep things dry, and they're using the stack capsules to do the high pressure, not the high pressure freezing, but the post high pressure freezing free substitution. And they're able to then use this in a correlative approach, the light microscopy before freezing, serial block face, XRM, correlative XRM, and serial block face. We see this region in here and this analysis. And an advantage that they describe here is they have a lossless handling of small plant samples. Plants float in water. Plants also, I understand, can float in inorganic solvents as well, depending upon where they are in the high pressure freezing. And by doing this entrapment, which can be done with the capsules at cryogenic temperatures, so a freshly high pressure frozen sample goes into the liquid acetone for the free substitution, and the buoyant plant specimens remain immersed throughout preparation because the fluid level is always above this height here where the two capsules are stacked. So it's an advantageous approach. They used a manual preparation, but an automated preparation could be done. They do not have an ASP. Probably one of the most clever and certainly in my case unanticipated applications for how the capsules could be used is shown in this particular work that came out of Madison Area Technical College and then later Ohio State. Cactus and kidney, and this is a cryo-SEM approach. They took one of the specimen capsules and literally sliced off the bottom part of the capsule, here shown on the top. And so they had a partial capsule in here. They then mounted specimens of cactus or kidney, fresh cactus or glutaraldehyde fixed kidney in the mPrep screen, slid the capsule down over this, and then removed it, having the specimen extend proud. So this is now a holder for the specimen in this location in here. These were then This is a photo of exactly what this looks like. This is a woody part of the specimen from the cactus. And this was then immersed into a cryogen. The cryogen, if I recall correctly, was liquid nitrogen slush. It may not be the most rapid freezing, but sufficient for this particular application. And then the wet specimen, because this was all done with the wet sample, was then clamped directly into the cryomicrotome. So this part of the specimen was filled with water, which is now ice, so they could clamp this directly into the cryomicrotome and cut a face across this to expose what the what the insides of, in this case, the cactus and the kidney look like. The samples were then freeze-dried. This is what they look like, again, diagrammatically. And the screen itself was used to manipulate the specimen, not the specimen, and placed on an SEM stub where it was conductive mounted and sputter coated, again, at room temperature following the freeze drying. And this is what we see here in this particular example. So you have these really informative cross sections that look like textbook drawings of what the tissues look like. In this case, Christmas cactus, which was fresh, and canine kidney, which was glutaraldehyde fixed and prepared in this way. And you have these absolutely stunningly gorgeous images. Another novel application is in biological safety, and this was work that was done by US AMRID. These researchers are working in a BSL-4 laboratory with really nasty virological agents in here. One can't imagine how difficult it would be to handle grids for grid-negative staining in this kind of environment. You can barely see the grid. You're working at a long working distance, and sharp devices are not allowed because you could puncture the suit and expose yourself to a pathogenic virus. So this was very, very difficult, and the success rate within this lab of being able to negatively stain these pathogenic viruses was quite low. I've been told levels of under 10%, maybe even 5% or so. But by loading filmed grids and placing them into the capsules, these scientists need only do the action of pipetting in order to deliver the viruses to the grid for negative staining. So they would, in the BSL 3/4 suite, they would pipette in the virus. They would simply then lay the pipetter down on its side for a period of time to allow sedimentation of the virus onto filmed grids, rinse and fix. And then they use one of two methods. They would take the capsules off, place them into a 50 ml centrifuge tube with an osmium tetroxide impregnated piece of filter paper, and they would then fix them and effectively deactivate the virus in the osmium vapor, or they would fill the whole chamber with glutaraldehyde. The glutaraldehyde, they'd have to sit in the glutaraldehyde solution for 24 hours. With the osmium vapor, if I recall correctly, it was simply 1 hour. Then they could take this out of the biocontainment suite, open this up, obviously in a fume hood, now in a BSL-2 EM laboratory, and do the stain and wash of the specimens. And here are some examples of some of the work that they showed. So we've got Ebola virus over here and chikungunya virus in this particular image here. So this solved a problem for them by being able to handle this and process these highly pathogenic specimens in a very reliable way. And in fact, the sample method was reliable enough and consistent enough that Candace Blanchett from this particular group was able to use this for quantification of virus counts by the ratio of the the number of virus images to colloidal golds that they were mixed with. And there's a citation in here if anyone wants to read on about that. So a very, very powerful technique that really eased their work and a very clever application. I'm going to conclude with the last application area where being able to do light microscopy and SEM with a 3D cell culture system. So not strictly EM, but this laboratory did some EM, and this was published and presented at Experimental Biology many years ago now. So in this particular case, a scaffold for 3D cell culture was entrapped in the capsule, and these little dots represent the cells. Now, one can use a light microscope looking through the holes in the capsule on the bottom and through the transparency of the capsule, and have a reasonably good, not a great quality, light microscope image. And this is bright field, nothing is stained in here, and one can make out the filaments of the scaffold in this particular image in here. But you could also image the scaffold from the side by simply laying it down in an inverted microscope and obtain pretty nice images, and again, bright field in this case. And you can see tumor nodules and colonies growing on the scaffold. Again, these were breast cancer cells. They're also controls, and it's a hyaluronic scaffold. And this was after a 20-day flow-through 3D tissue culture in the capsule. So I'm not showing the flow-through system that was used during the culture in here. And one can, again, just simply through the side of the capsule, do some reasonable bright field imaging of this. And one could imagine that fluorescence and other kinds of probes could improve this. These can then be prepared for SEM. So one can do a correlative approach in the SEM. And one can also collect the cells that come out through the capsule and run flow cytometry and do some analysis on the cells there. And in this particular case, the breast cancer cell scaffolds had a cancer-like phenotype, but I'm not showing all the data from this. So that kind of shows a breadth of ranges. I very rapidly explored a range of specimens: brain, nerves, retina, kidney, muscle, fish gills, tumor tissue. I showed some plant examples and obviously some viruses, a variety of methods that could be done that show some of this versatility: volume EM, TEM, SEM, light microscopy, XRM, cell pellets, cell culture, and even an example of freeze substitution. There was a lot I have not shown you. I haven't shown by any means the breadth of mammalian tissues or non-mammalian tissues, and in particular, I haven't shown some of the other methods like multi-parameter preparation tests that, for example, Melania McLean, Kader Narayan, and Kurt Zimick use with the system in order to develop which is the best embedding resin or the best staining protocol or some others. I haven't shown spheroids. I haven't shown acrylic resins. I haven't shown cover slips. And I obviously have not shown other specimens, in this case, sort of more materials. So with that, I'd like to acknowledge the customers and collaborators and some of the work that was shown today. There's quite a few here and many, many others that are involved in other studies that we like to support and help enable what we can with a system that we hope is adaptable and, as demonstrated here, can be MacGyvered into some novel applications as well. So with that, I'm happy to take any questions. My email is here as well. I'm sure you can all find me.